morning, students. Today we start with the proteins and how the proteins are formed and chemical nature of proteins and structure of proteins and denaturation. Okay, listen. Here, first we may take that proteins. And here we often, this corona time, we may hear the word proteins. Most of our doctors suggested take protein food. Now, chemically, what is protein? So, proteins are polymers of alpha amino acids which contains peptide bond. So, we are taking chemically, proteins are polymers of alpha amino acids. Alpha amino acids which contains peptide bond. Which contain peptide bond. Contains peptide bond. What is it? Peptide bond. Listen. Here, what is it? Peptide bond. So we are taking chemically what is protein. Proteins are alpha amino acids which contains peptide bond. Okay. Now, what is the peptide bond? Listen. It is peptide bond. Peptide bond. Peptide bond is nothing but amide bond. So what is it? It is amide bond. So it is amide. So amide bond is considered as a peptide bond. So listen, what is it amide? It is C double bond O and NH. So C double bond O and that NH bond, N and that H. This bond is considered as, that is the amide bond. So what is that? Amide bond. So all the peptide bond which consists of amide. So what is that amide? CONH group is considered as amide. And how will you form this amide bond? So listen. How will you form your amide? So, we are taking amide bond or peptide bond is formed from alpha amino acids. So, these are polymers of alpha amino acid. From that amino acid, we make a amide bond. So, here the peptide bond or amide bond, it is formed from alpha amino acids. Alpha amino acids. What is it? First, alpha. Alpha means next to the functional group. Next to the functional group, which is known as alpha. And here amino. Here amino means it's nothing but NH2 group. We are considered as amino. And acid, here the acid. What is that acid group? Here it is C double bond O and that OH. This is an acid. So what do you mean by alpha first? Alpha means next to functional group. So this is COOH is a functional group. Next to functional group, there is a carbon, then it is known as alpha carbon, and after that the NH2 group. So we are taking that amino acids. So we are taking the amino acids, alpha amino acids. So listen here, that is alpha amino acids. Alpha amino acids. Here C double bond O, OH. It is acid group. Correct? Then next to that acid group there is CH. Then it is alpha carbon. So it is alpha. And in that alpha, the NH2 group is present. Here there is NH2 group is present. So alpha carbon which contains NH2 group, so that it is known as alpha amino acid. And here the acid is a functional group and NH2 group is a substituent, and here there is a R group. What is that? R group. Depending upon the R group, we are getting so many amino acids. So this is the amino acids. What is that? It is a amino acid. Amino acids. So what is amino acid? It is alpha amino acid because amino group is present at alpha carbon. Now here we are getting that the CONH bond. What is that CONH bond? We have previously taken this peptide bond. So what is that peptide bond? It is a CONH bond. How will you get this peptide bond from that amino acid? Now, one amino acid, the NH2 group of one amino acid is reacted with the COOH group of other amino acid, then it will be leads to a peptide bond. So listen, here we are getting that a peptide bond. So it is a peptide bond. Now alpha amino acid is taken here. Then here another one COOH and NH2. So here it is reacted with one more amino acid. This is NH2. It is NH2, and here we are taking that is a CH and that R and COOH. COOH. It is C double bond O and that OH group. Now we are taking that two amino acids. It is the first amino acid and it is the second amino acids. Now these two are reacted. How they are reacted? Is a NH2 group of one amino acid is reacted with the COOH group of another one amino acid. So that here the OH group of one amino acid is reacted with one of the hydrogen of the other amino acid, that will be leads to peptide bond. So that we are getting a peptide linkage. So here it is NH2 bond CH and that R, and here it is CONH. Listen, here C double bond O and that NH. This is a peptide bond. So CONH and remaining it is CH and here R and COOH. Okay. Listen, here it is the peptide bond. So we are getting a peptide bond, and peptide bond is nothing but CONH bond. So CONH bond is nothing but peptide bond. So we are getting this peptide bond, that is CONH bond. CONH. Now we are calling this peptide bond. And what is the peptide bond? How will you form? We are getting two amino acids. Then two amino acids are reacted together by removal of water. We are getting a peptide. Now this peptide we may call this a dipeptide. What is that? Dipeptide. Why we may call this di? Because number of amino acid joining is nothing but 2. So here 1 amino acid and here 2 amino acid. So 2 amino acids are joined and form a peptide which is known as dipeptide. Listen. Here a di. What do you mean by di or bi in chemistry? Di means 2. Mono or uni, that means 1. Tri means 3. Now, di means 2. This di, which means 2, which represents 2 amino acids. 2 amino acids. So, there may be a question, a dipeptide, which contains how many peptide linkage? A dipeptide, which contains only 1 peptide bond. Correct? So, if we are taking one more example, if we are taking a tripeptide. Listen. So, what do you mean by tripeptide? It should be 3 amino acids are joined. So, we are taking that 3 amino acids. Here, NH2. It is amino group, and it is amino acid, that is CH, and here R. And it is COOH, that is C double bond O, and that it is 1 amino acids. 1 amino acids. Okay, so this is the 1 amino acid is reacted with another 1 amino acid, that is NH2, NH2 bond, and here it is CH and R, and here it is C double bond O and that OH. It is second amino acids. Now it is reacted with the third one. What is that? It is reacted with the third amino acid. What is that? Here it is NH2, and here it is CH alpha carbon and that R, and that here it is COOH. It is COOH group. So we are taking 3 amino acids. We are taking 3 amino acids. What are they? Here it is 1, and it is 2, and that it is 3. So we are taking 3 amino acids. Now 3 amino acids are joined together, and NH2 group of 1 amino acid is reacted with COOH group of another 1 amino acid and make a peptide bond. Now we are get this NH2. We are taking the product. It is NH2, and here bond CH bond R, and here there is a formation of peptide bond. So here it is C double bond O and the NH. So that these two are reacted. So the OH group and the hydrogen is reacted by removal of water. We are getting 1 peptide bond here and remaining. So what is that? Here it is CH and R. And remaining, what is that? Here again the CO and the NH2 group is reacted and form a peptide bond. So that here it is a C double bond O and that NH. Correct? And this is the OH group and the hydrogen is reacted together and form a peptide bond. And remaining, so what is that? Here this is the CH and that R group. And here we are taking that is a COOH group. So the third amino acid, the COOH group. Now there are 3 amino acids are joined together. 3 amino acids are joined together. The first one and second one and the third one. Now we may get a peptide. The peptide is known as tripeptide. What is that? Tripeptide. So this is a peptide bond. What do you mean by peptide? Peptide is nothing but CONH group. Now, what represents this tri? The tri represents, it will be formed formed by 3 amino acids. There is 1, under that it is 2, under that 3. So 3 amino acids are joined together. We make a tripeptide. So what is that? Tripeptide. So a tripeptide which contains how many peptide linkage? So a tripeptide which contains 2 peptide linkage. 2 peptide linkage, correct? So the CONH group is nothing but the peptide linkage, so that it contains 2 peptide linkage. So they are asking in place, a tripeptide contains 2 peptide linkage or 3 peptide linkage or 4 peptide linkage like this. And most of the students are misunderstanding this tri means 3, so that it contains 3 peptide linkages like this. But the tri represents number of amino acid combined together. So if we are taking a tripeptide which contains only 2 peptide linkage, and here if it is a dipeptide, then it is only 1 peptide linkage. So generally we may call here, that is n number of amino acids are joined together, then n minus 1 peptide bond is formed. So we are taking n amino acids are joined. So we are taking n amino acids. N amino acids are reacted together. N amino acids is reacted together and form n minus 1 peptide linkages. That is n minus 1 peptide bond. So that n minus 1 peptide bond. So we are getting the n minus 1 peptide bond. Now we may take the topic. What is that? It is protein. What is a protein? Proteins are alpha amino acids, the polymers of alpha amino acids. So we are taking generally proteins are polymers of alpha amino acids. So we are getting a polypeptide. So a dipeptide, a tetrapeptide, and the polypeptide. So proteins are considered as the polypeptide. Now, if we are taken 3 amino acids, we are getting a tripeptide. So we may take that if 20 amino acids are joined together. So we are taking if 20 amino acids. 20 amino acids. So 2 to 20 amino acids are present in the or form a peptide then which is known as oligopeptide. They are called as oligopeptide. So what is it? Oligopeptide. Oligopeptide is nothing but 2 to 10 or 20 amino acids are joined. We may get the oligopeptide. Now, more than 20 amino acids. More than 20 amino acids. So we are taking more than 20 amino acids. So more than 20 amino acids, that will be leads to polypeptide. That will be leads to polypeptide. So here proteins are chemically polypeptide. So if you are taking a protein, listen here, if you are taking a protein, proteins are polypeptide. That is more than 20 amino acids. That means here most probably, that is more than 100 amino acids are joined, then we may get a protein. So in protein, here it has more than more than 100 amino acids. More than 100 amino acids are joined together. We may get protein, and also its molecular mass, the molecular mass is greater than 10,000 units. Greater than 10,000 units. So proteins are polypeptide. Proteins are polypeptide. So do you know that here most of the toxins we are taking that do you know that we are taking that? Here, most of the toxins in animals. So we are taking most of toxins. Most of the toxins. Most of the toxins. Most of the toxins in animal. Most of the toxins in animal. And also the plant venoms. Plant venoms. So plant venoms all are proteins. So all are, that is, proteins. So if you are taking the proteins chemically, these are the polymers of amino acids, and here we are taking most of the toxins in animals and the plant venoms all are, that is, proteins. If you are taking the substituted for sugar, we are taking aspartame. What is that? Aspartame. It is aspartame, a chemical which is used to substitute for sugar. So it is the substitute for sugar. So it is substituted for sugar. And this aspartame is a dipeptide. So aspartame is a dipeptide. So we are taking the aspartame. It is a dipeptide. Suppose we may take the structure of aspartame. Listen. The structure of aspartame, that is NH2. That is, two amino acids are joined together. We are taking that. That is aspartame. So we are taking the aspartame. That is NH2. And here it is CH. And we are taking in alpha carbon. It is CH2 COOH. Instead of R group, that is CH2 COOH. And then here that is CONH group. That it is peptide linkage, that is CONH group. Listen, it is CONH group. CO under that NH, that is peptide linkage, and after that it is CH. So here it is alpha carbon of another one amino acids, and here it is CH2, then C6H5, it is phenyl alanine group, phenyl alanine group, and then it is COOCH3, COOCH3. Then it is a aspartame which is used to substitute for sugar like that saccharide. Okay, and it is 100 times greater sweetest taste than that of sucrose. So this aspartame is 100 times sweeter than sucrose. 100 times sweeter than sucrose. So it is 100 times sweeter than sucrose. So the aspartame, it is diastolic, right? Now we are taking the structure of protein. So let us discuss here the structure of protein. So here we are taking the protein. So what is a protein? Proteins are polymers of amino acids. So proteins are polypeptide. So listen here once again, what it is protein. Proteins are polymers of alpha amino acids. And alpha amino So what is a peptide bond? Which contains CONH group, which is known as peptide bond. And how will you form the peptide bond? And it is formed from alpha amino acids. Alpha amino acids. And alpha means next to functional group, and amino group means NH2, and acid means COOH. Okay. Listen, here if you are taking an alpha amino acid, two alpha amino acids is joined together and form that a dipeptide. A dipeptide which contains only one peptide bond. Now, if you take three amino acids, that is 1 and 2 and 3, three amino acids are joined together, we are getting that is a tripeptide, and a tripeptide which contains two peptide linkage. So, if n amino acids joined together and form the n-1 peptide bond. Now, if 2 to 20 amino acids are joined, then we may call this oligosaccharide or oligopeptide. Now, more than 20 amino acids are joined, we are getting that it is a polypeptide. Now, proteins are considered as more than 100 amino acids. So, more than 100 amino acids, that means it is a polypeptide, and its molecular mass is greater than 10,000 units. Okay, so most of the toxins are the poisons in animals, and also that the plant venoms are proteins. Now, if you are taking an aspartame, it is also a protein which consists of only 2 amino acids. So that we are called as it is a dipeptide. Dipeptide. Now, it has 100 times sweeter than the top sucrose. Now, we may discuss about the structure of protein. So that we are taking the structure of protein. Structure of proteins. And here, the structure of proteins, we are taking that 4 levels of structures. So that the first one, it is primary structure. So what is it? It is the primary structure. So we are taking primary structure. Primary structure. So we are taking the primary structure and secondary structure and tertiary and the quaternary. So that primary structure and that secondary structure, then tertiary structure, and the fourth one, that is quaternary structures. Quaternary structures. So, the structure of proteins we may consider as 4 levels. One is primary structure, and the secondary, and tertiary, and the quaternary structures. So, the first one we are taking, that is the primary structure. So that we are taking the primary structure. So, listen, we are taking the primary structure of protein. So, here it is primary structure of protein. Listen. So, the primary structure of protein, which refers number of amino acid and the sequence. So, the primary structure which refers number of amino acids, and its sequence. So, number of amino acids and its sequence. So, number of amino acids and the sequence is represented by this primary structure. And here, if you are taking if there is a small change in sequence, then will alter the overall structure and the biological function of protein. So, if there is small changes in sequence, small change in sequence, small change in sequence will alter the structure and the function of protein. So it will alter the structure and function of proteins. So the structure and the function of proteins is dependent on primary structures. Structure and function of protein, that will be dependent the primary structure. Suppose if you are taking the hemoglobin, we are taking the hemoglobin. So listen. So we are taking the primary structure. Primary structure refers number of amino acids and its sequence. Now, small change in sequence will alter the structure and the function of proteins. If we are taking that hemoglobin, so here we are taking a hemoglobin. Hemoglobin. What is a hemoglobin? It is a red blood cell which carry oxygen from lungs to tissues. So this is the function of hemoglobin. So it is a hemoglobin. So we are taking a hemoglobin, and in this hemoglobin we are taking, listen here, the normal hemoglobin sequence is, we are taking the normal sequence. Normal sequence. So normal sequence. So normal sequence in hemoglobin. Normal sequence in hemoglobin, we are taking, listen, we are taking that valine, we are taking valine, and it is histidine, valine, histidine, and leucine, and threonine, leucine, and threonine, that is THR, threonine, and that is proline, that is proline, and that is glutamic acids. So here it is glutamic acid, and another one it is glutamic acid, and another one it is lysine. It is lysine. So the normal sequence in hemoglobin is valine, histidine, leucine, threonine, and proline, glutamic acid, and the lysine. Here, if you are change the sequence, what will happen? So listen, if you are change the sequence, here we are taking that the glutamic acid is there. So listen, here there is a glutamic acid. And instead of glutamic acid, we are taking that is valine. So we are taking, it is valine. So what is that? It is valine, and it is histidine, and it is leucine, and here it is threonine, that is THR, and it is proline. Here it is the proline. Then here instead of glutamic acid, we are taking that is valine. Here in the above we are taking glutamic acid, and here there is a valine. And again it is glutamic acid. So again it is the glutamic acid, and again it is lysine. Again it is lysine. Now, what will happen here? There is a small change. What is it? The sequence is very small change. One of the amino acid in normal hemoglobin, there is a glutamic acid. And if you change that, glutamic acid, we are taking with the valine, then what will happen? It will be loses the biological activity of hemoglobin. What is the function of hemoglobin? That is, it will be carries oxygen from lungs to tissues. The loss of oxygen carrying capacity is introduced by taking the sequence or change the sequence. So, if we change the sequence, then we may get it is sickle cell anemia. So, we are taking it is the sickle cell anemia. Sickle cell anemia. So, the sickle cell anemia that will be created while changing the sequence, that is, hemoglobin lose the capacity to carry oxygen from lungs to tissues. So, primary structure is very, very important, and we cannot change the sequence. Suppose if you are taking the sequence or change the sequence, it alter the structure and the function of proteins. Structure and the function of proteins. Here, if you are taking the sequence of amino acids, So, we are taking in that diagram, we are taking that the sequence of amino acids in some cases. This is the sequence of amino acids. Listen here. Now consider the sequence or peptide bonds in amino acids. So here we are taking so many sequences. So here lysine, valine, and that is phenylalanine, glycine, all are the sequence of amino acids. And if we change the sequence here, what will happen? Here that will be cause the diseases. Here instead of if you are taking that instead of arginine, we are taking that here there is a arginine present. Instead of arginine, we may take the valine. Then it will be alter the function, biological function, and also the structure. So it is completely different protein or we make it. So the sequence that will be represents, that is a primary structure. So primary structure represents the sequence of amino acid. If there is a small change in the sequence, then there is a change in structure and also the biological function. Example, we may take that the hemoglobin, it has normal oxygen carrying capacity. And after that, if we are taking the glutamic acid, instead of glutamic acid, we are taking the valine, then what will happen? We are taking sickle cell anemia. It will cause some diseases. So here also there is a sequence. So we may take that the next one, it is a secondary structure. So we are taking the secondary structures of amino acids. So listen. So we are taking that secondary structure of amino acids. So the second one, it is secondary structure. Secondary structure. And what refers to the secondary structure? The secondary structure refers, so we are taking the secondary structure, the manner of protein chain which is folded. So the secondary structure that will be refers the manner that will be represented by secondary structure. So the manner of protein chain folded or bent that will be represented by the secondary structure of protein. And another one, we are taking that the second one here, the what is the chemical nature or chemical bond present in amino acid that will be explained by the secondary structure. So nature of bond, we are taking it is the nature of bond. So here we are taking the nature of bond. Nature of bond. So nature of bond which stabilizes the protein molecule will be explained by this secondary structure. So secondary structure which represents manner of protein chain which is folded and nature of bond. Now, if you are taking the manner of protein chain bonded, there are two types. One is alpha helix. Alpha helix. So here it is alpha helix, and the second one it is beta pleated. So it is beta pleated sheet. So alpha helix and beta pleated sheet that will be represented, that is the manner of protein chain that will be folded. So there are two manner, one is alpha helix and then the beta pleated. And so first one we are taking that is the alpha helix structures of protein. Alpha helix structures of protein. So listen, it is alpha helix structure of protein. So we are taking that is alpha helix structures. Alpha helix structure of protein. Alpha helix structure of protein. Okay. Now we may take that they are found away, that is the alpha amino acids which are coiled in the right-handed screw. So all alpha amino acids we are taking here. Alpha helix structure is formed by coiling of alpha amino acids. So they are formed by coiling of alpha amino acids. Coiling of alpha amino acids. Coiling of alpha amino acids by right-handed screw. So by or as right-handed screw. They are formed by likewise, that is a right-handed screw. Right-handed screw. So here we are taking the alpha helix structure. So the alpha helix structure is formed by coiling of alpha amino acid as right-handed screw. Right-handed screw. So we are taking that like a right-handed screw. Listen. Here it is right-handed. Right-handed screw. So they are formed by coiling of alpha amino acids. So alpha amino acids are coiled as right-handed screw, then we may get the alpha helix structure. And why they are coiling? They are coiling because of hydrogen bond. So this is because of hydrogen bond. So because of hydrogen bond, they are coiled. And how the hydrogen bond is present in alpha helix. So here, how the hydrogen bond is present in that alpha helix. That means here we may take that the one unit we are taking the NH bond bond of one unit of amino acid, NH bond of one unit is making a bond, is making a hydrogen bond with fifth unit. With fifth unit of carbonyl CO bond. So here the hydrogen bond is formed between one unit of NH bond to fifth unit of this CO bond, so that they are making the hydrogen bond. So due to this hydrogen bond, we are taking that this hydrogen bond is formed between amino acids one unit to the fifth unit, that is N2. Here one unit is considered as N, and it is considered as N. One unit is considered as N. Then the fifth unit is considered as, it is fifth unit, it is considered as N plus 4. N plus 4 unit. So they are making a hydrogen bond with N and N plus 4 unit. Now, here due to this hydrogen bond, the alpha helix structure is holding in position. So due to this hydrogen bond, alpha helix structure is hold in position. Alpha helix structure is hold in position and stabilize the structure. So which stabilizes the structure. So which one which stabilize the alpha helix structure? That means that is a hydrogen bond. So hydrogen bond which stabilizes the structure. So we are taking the alpha helix structure. Alpha helix structure is formed by coiling of alpha amino acids, and it is due to making a hydrogen bond with the N and the N plus 4. Due to hydrogen bond, we will stabilize the alpha helix structure. So it will be stabilizes the alpha helix structures. And we are taking that wool and that and we are taking that is wool and that air are example of alpha helix structure. So alpha helix structure, we are taking an example wool and that air. So these are example of alpha helix structure. And we are taking alpha helix structure, and if the structure, the hydrogen bond is breaking. Suppose we are taking the hydrogen bond. So we are taking the alpha helix structure. Here it is an elastic nature. It is elastic nature. How it is elastic? If we are stretched, then what will happen? The hydrogen bond form is breaking. Then we may release the tension, the hydrogen bond is reformed, so that it is elastic nature. It is an elastic nature. So hydrogen bond alone, there is an elastic nature. If we stretched, then hydrogen bond is breaking. Hydrogen bond is breaking. So hydrogen bond is breaking. If we release the tension, there is a reformation of hydrogen bond. If we release the tension, hydrogen bond is reformed. Hydrogen bond is reformed. So these are elastic nature. This is elastic nature. So hydrogen bond is reformed so that it is elastic nature. Okay. Now, in each one helical structure, each term which consists of 3.6 amino acids. So, each term, there are 3.6 amino acids, 3.6 amino acids which present in each term, and its bond length is equal to 5.4 angstrom. 5.4 angstrom. So this is the secondary alpha helical structure. So the secondary alpha helical structure we are taking. The secondary alpha helical structure is formed by coiling of alpha amino acids as right and S2. And here, because of hydrogen bond, there is a coiling occur. Because of hydrogen bond, there is coiling occur. And the NH bond of one unit is making hydrogen bond with the fifth unit of CO bond. And due to hydrogen bond, alpha helix structure is hold this position and stabilize the structure. Wool and air are alpha helical structure. And it is elastic nature. If we stretch, hydrogen bond is breaking. Then, if we release the tension, the hydrogen bond is reformed. So, each term which consists of 3.6 alpha amino acids and the bond length is 5.4 angstrom. Now, if you have seen that here, the alpha helical structure. So, listen, the alpha helical structure will be taken a diagram. Alpha helical structure. So this is the alpha helical structure. So listen, this is the alpha helical structure. So this is the alpha helical structures of amino acids. Alpha helical structure. And here there is the formation of hydrogen bond. Listen, here the NH bond. What is that? Here there is the NH bond. So CO and NH bond. So here there is a CO bond, and here we are taking the NH bond. So NH bond and the CO bond is NH and the CO bond is making the hydrogen bond. So this is the hydrogen bond between that is the amino acids. And if you are considered it is considered as a N, then what will happen here? The NH position should be N plus 4 unit. So first and fifth unit making a hydrogen bond and in the protein, and here each term which contains 3.6 amino acid in each term. So the amino acid in each term which contains that is 3.6 amino acids. So this is the alpha helical structure of amino acid. Alpha helical structure of amino acids. Now we are taking beta pleated structure. Beta pleated structure. So we are taking another one secondary structure. It is beta pleated structure. Beta pleated structure. Now we are taking the beta pleated structure. Okay. The next structure we are taking that is beta pleated structure. Beta pleated structure. So the secondary structure, it is beta pleated sheet. Beta pleated sheet structure. So the beta pleated sheet structure, is the secondary structure. It is the secondary structure of protein. Another one type of secondary structure is beta beta pleated sheet. And what will happen in beta pleated sheets? Here, the amino acid chain is arranged in side by side. So, if you are taking the amino acid, so amino acid chain we are taken. So, the amino acid chains are arranged. Amino acid chains are arranged in side by side manner. Side by side. So, side by side manner, then we are getting that beta pleated sheets. We are getting the beta pleated sheets. And here, we are getting large number of hydrogen bond is formed. So, very large number of hydrogen bond is formed in beta pleated structure. So, very large number of hydrogen bond is formed in beta pleated structures. And here, the sheets are slide over each other and forming a three-dimensional structure. So that the sheets, we are taking the beta pleated sheet. So the sheets are slide over each other. So the sheets are slide over each other and forming the three-dimensional structure. And we are taking an example here. We are taking the beta pleated structure. That is example. It is the silk. We are taking the silk. Now we are taking the beta pleated structure. So we may take the diagram. This is the beta pleated structure. This is the beta pleated structures. Beta pleated structure. And alpha helix structure. So here each and every one is slide over and we may get the three-dimensional structure. So here more number of hydrogen bond is formed in beta pleated structure and between NH and CO bond. So listen student, this is beta pleated. So here it is a beta pleated structure. In this beta pleated structure, we may get more number of hydrogen bond is formed between NH. So here there is a NH and the CO and the CO and the NH, and here CO and the NH. There is a formation of hydrogen bond. And here CO and the NH. So more number of hydrogen bond is formed between two amino acids or more than amino acid units. So that this is the beta pleated structure. So it is beta pleated structure of amino acids and proteins. Now, this is the alpha helix structure. Compare that alpha helix structure and that beta pleated structure. Number of hydrogen bond is maximum in the case of that is beta pleated structure. And example of this alpha helix, we are taking the example. What are the example? We are taking that is air and the wool. Air and wool, these are example of alpha helix. And beta pleated, then we are taking that is a silk. What is the difference? Here, it has right-handed coil. Alpha helix structure is right-handed coil. And beta pleated, it is slide over and over and making that the three-dimensional structure. So that here we are taking the tertiary structure of protein. So the next we are taking the tertiary structure of protein. So we may consider the primary and the secondary structure. Now we are taking the tertiary structure. So that we are taking the tertiary structure of proteins. So listen. So that the third one, it is the tertiary structure. Tertiary structure of protein. So that we may take the tertiary structure, and here the tertiary structure represents a further folding of a secondary structure. So that the tertiary structure represents further folding of folding of secondary structure which represents, that is tertiary structure. So what will happen here? The secondary structure, it consists of alpha helix and beta pleated. So beta pleated and alpha helix structures are taken as their secondary structure, and it will be further folded on, that is we make a tertiary structure. And here in a secondary structure we are taking only the hydrogen bond. So besides hydrogen bond, there are so many forces will act as stabilizers this tertiary structure. So here this tertiary structure will stabilize us. So this tertiary structure will stabilize by one is hydrogen bond, that is hydrogen bond between CO and NH group, and the second one, it is electrostatic interaction. So electrostatic interaction, that will be stabilizes the tertiary structure, and the third one, it is hydrophobic interaction. What is that hydrophobic interaction? Because of R, so this is the hydrophobic interaction. Hydrophobic interaction in amino acids, there will be R group, then it is considered as the systemic clearance. So hydrophobic interaction, and also that is sulfur bridges. So these are the forces that will be stabilizes the tertiary structure. So sulfur bridges. So here we may take an example. Here we are taking the insulin. You know that insulin. What is the use of insulin? It will be control the sugar level. So we are taking insulin. Insulin which control the sugar level in blood. So which control sugar level in blood. So insulin which controls the sugar level in blood and which consists of the sulfur bridges. So insulin which contains sulfur bridge. So sulfur linkages which stabilizes the tertiary structure of insulin. Now we are taking the quaternary structure. So we are taking the quaternary structure. So we are taking the quaternary structure. So the fourth structure, it is quaternary structure of protein. Here, some amino acids, we are taking some amino acids which contains more than one polypeptide chains. So, some amino acids which contain are made up of more than one polypeptide chain. So, if you are taking some amino acids, some peptides which contain more than one polypeptide chain. One polypeptide chain. One polypeptide chain. And this spatial arrangement of more than one polypeptide chain is refers as the quaternary structure of protein. And here, suppose if you are taking the hemoglobin, that is hemoglobin. Hemoglobin. And hemoglobin, which consists of 4 polypeptide chain. 4 polypeptides. Polypeptide chain. So, hemoglobin, which consists of 4 polypeptide chain. 4 polypeptide chain, and the interaction between these 4 polypeptide chain that will be represents the quaternary structure of protein. And also, if you are taking that, the DNA polymerase. So, DNA polymerase. So, the DNA polymerase which contains, that is 10 polypeptide chain. 10 polypeptide. So, this is 10 polypeptide chain, and this folding of these 10 polypeptide chain that will be represented by this quaternary structure. So, that quaternary structure represents folding of more than one polypeptide chain. So, here quaternary structure that represents folding of more polypeptide chain. More polypeptide chain. Folding of more polypeptide chain that will be represented by these quaternary structures. Quaternary structure. Now, if you are taking the both primary and secondary and tertiary structures and quaternary structures of protein. So, if you are taking a diagram here, listen. Okay, this is a diagram which is listen. Here we are taking all those primary and secondary and tertiary and quaternary structures of protein. So listen, if you are taking the primary structure. So here, listen. So if you are taking all those structures, we are taking primary, secondary and tertiary and quaternary structures of protein. And primary structure that represents sequence of amino acids, and secondary structure they are taking the primary structure and the secondary structure and the tertiary and the quaternary structures. There is a primary structure that will be represents sequence of amino acid. What is the cause of sequence of amino acid? If we may change the sequence, then it will be alter the structure and biological function of amino acids. So this is the primary structure that represents amino acids. Okay, and if we change any one amino acids into another one, then the biological function is valid. Then we are taking the secondary structure. If you are taking the secondary structure of protein, it classified into two types. One is alpha helix. It is alpha helix. So it is an alpha helix structure and it is beta pleated. It is beta pleated structure. So here alpha helix and beta pleated structure, then it will be stabilizes the secondary structure of protein. And tertiary structure, folding of secondary structure, that will be represent the tertiary structure. And here besides the hydrogen bond, there is electrostatic force and also sulfur linkages in insulin, and also we are taken hydrophobic interaction stabilizes the stress structure. And coordinate structure, more polypeptide chain are linked together, we may get the coordinate structure. We are getting the coordinate structure. So primary structure means sequence, secondary structure hydrogen bond, and tertiary structure, that is three-dimensional folding of secondary structure, that will be reached to, that is tertiary structure. And quaternary structure, folding of more than one polypeptide chain, that will be represents, that is quaternary structure. So these are the structure of protein. Students, go through your textbook and CBSE book, then thorough it, and hope you will learn the maths. Thank you.